Hey physiology students, welcome to Wednesday's lecture. This is going to be the last of chapter 2, part 2, cell transport, and today we'll talk about active transport, and it won't be a long lecture. So first I do want to go to my website, and I'm going to share the screen here in just a second. There we go, share screen. Want to keep you updated as to what's going on and to remind you about the format for not this coming week that you're watching these lectures, but the following week when we're supposed to return to campus. Okay, so let's make this a little bit bigger so everyone can see it. Okay, so we are, let's see. Okay, so this is Wednesday's lecture, which will be September 2nd. Okay, so we're still online, as you can see right here, and we'll go through chapter 2, part 2. Now, Friday's lecture during this week is going to fall on September 4th, but since we're finishing chapter 2, part 2 today, we'll start chapter 4, part 1 on this Friday, September 4th, okay? And September 7th, we'll continue on with this material in chapter 4. Now, starting September 9th, okay? Starting September 9th, which will fall on a Wednesday, all right? That is when we're supposed to be back in class. And this is when we'll be in the University Center, the UC Gymnasium North, okay? So I recommend you find out where that is before we're meeting in person on September 9th. All right, so that is the format. If anyone has any questions, don't hesitate to email me with those questions. All right. Okay, another thing I want to remind you of is Friday, September 4th, we'll have our first lecture quiz. It's already up. In fact, someone in class has already completed it and turned it in, but that will be due by 12 p.m. on that Friday. All right, so you fill it out, email it back to me. Okay, all right, so let's stop this share and let's go to our PowerPoint where we want to finish this material. Okay, so we had gone through passive transport, okay, and that leaves us with active transport today. All right, so please make note that when I have the page number for the wiki text, you can go to the wiki textbook, the PDF, type that page number in, and you'll be brought right to that section where I talk about that, okay? So, for example, let's do this. I'm going to go to my document. I'll find it here. Okay, here is my document. Okay, so I'm going to make this a little bigger for you. You'll never be able to see it. Okay, page 1 right here. Now, in my PowerPoint, it said page 17 will bring you to where it talks about active transport. So let's put that in there. Page 17. Bingo. There you are. Active transport. Okay. I talk about ion pumps, and I have my little dialog box down here. And if you go down further, I say, oh, look, skip this part. It's bulk transport. Well, actually, don't skip this part. You do want to know this. We'll cover this, okay? But let's see what else. Parts of a cell. Here's where I say, skip this section, okay? You don't have to worry about that because I don't cover it in my chapter, okay? And I'm seeing if in the chapter there's anything else that you need to know. And see, pretty much it's just the beginning of the chapter that you'll have to know. Cell metabolism, we've already covered. All right, that was chapter 2, part 1. Okay, but I did want to let you know about that page number thing because that's important. Okay, so back to active transport. This is going to be moving against the concentration gradient. So this is moving substances where they're in low concentration across the membrane to where they're in high concentration, and that's going against the current. That's going to take energy or ATP. So the first type of active transport, these are all going to be pumps. They're ATP-fueled pumps that pump things across the membrane. So calcium pumps. Calcium is normally low inside of a cell, and we'll find out in chapter 4, part 1, that calcium is an ion that can stimulate a cell, and so cells are very careful about how much calcium they'll allow inside. Okay, calcium pumps are important in muscle cells, and particularly cardiac muscle cells and smooth muscle cells smooth muscle you have in your GI tract. You have smooth muscle in the bronchioles of your lungs. You have smooth muscle lining your urethra, your bladder, um, also lining your arteries, which allows them to vasoconstrict or dilate. Okay. All right. So calcium pumps are important in muscle cells. Hydrogen pumps, proton pumps. If you remember when we talked about in chapter 1 the importance of pH, and I said pH is determined by the presence of hydrogen ions, which makes things more acidic, versus hydroxide ions, which make things more alkaline. So proton pumps are used to increase acidity, and the best example of this is your stomach. Your stomach contains hydro, or the hydrogen, chloric acid, and we have parietal cells in our stomach that are proton pumps that pump out the hydrogen ions to make your hydrochloric acid in your stomach. Now, this is a good writing assignment. Remember, we have a lecture writing assignment. Some people are prone to gastroesophageal reflux disease. Gastro is for the stomach, gastro. Esophageal means your esophagus, and reflux means something's moving in the opposite way it should. Now, your esophagus meets with your stomach at the cardiac sphincter. That sphincter is supposed to close off so that acid and other things from your stomach don't back up past that sphincter and get into the esophagus. Now, some people with gastroesophageal reflux disease, or GERD, might have a weak cardiac sphincter. They might produce too much acid. One problem or another, whatever it is, acid backs up from the stomach into the esophagus, and it causes pain. All right, people notice this at night when they lay down, and they're more prone to feeling that pain of gastric reflux. Now, the problem with gastric reflux that is left untreated is that that acid is going to erode a hole in the esophagus. It's going to first cause an ulcer, which can cause bleeding, and left untreated, eventually that acid will digest basically through the wall of the esophagus, leading to a perforation, which is bad, because now you have gastric contents that get into your thoracic cavity. Now, people sometimes go to the hospital because they're having chest pains, and they say, oh, I might be having a heart attack. Well, you'll examine them to see if they're having a heart attack, and if the EKG comes normal and the protein tests, the blood tests come back normal, and they find no indication of a heart attack, they might ask the person, do you have a problem with gastric reflux disease? And the patient goes, yeah, how did you know? Because the esophagus runs posterior to the heart. And when you feel pain in your esophagus, some people think that's referred pain, that they're feeling it in their heart. Okay, so are these pumps important in human health? They sure are. Gastroesophageal reflux disease must be treated so that you don't get a hole in your esophagus. Why doesn't the stomach digest itself with acid? Because the stomach produces a ton of mucus to protect the stomach from being affected by the hydrochloric acid. Your esophagus doesn't have that kind of mucus, so it's vulnerable. One treatment for GERD is to take Nexium, the purple pill. And Nexium is a proton pump inhibitor, okay? And what it does is it slows down those proton pumps in the stomach, so not so much acid is being produced, so it's less likely to reflux into the esophagus. All right, so here's active transport, proton pumps. Here's a case where there's human health as a result of these proton pumps being a little too excitable, okay? All right, last pump that I'm going to talk about that works by active transport is very important. The sodium-potassium pump. All right, we'll end this chapter talking about the importance of that. So what happens is sodium is like calcium. It's very stimulatory on a cell. So normally a cell keeps levels of either calcium or sodium outside the cell, and it keeps low levels of sodium inside of a cell because it's going to be stimulating a cell if it's inside. So, and potassium is important for relaxing a cell, okay? So it's important. A cell wants to hold on to potassium, but keeps a lot of sodium out, okay? So here's what this pump does. For every 3 sodium it kicks out of the cell will take in 2 potassium. All right, so if we look at the little GIF here, 3 sodium are being ejected from the cell, and then 2 potassium will be brought back in. All right, well, they're showing There is the potassium, the little blue K's are being pulled in. 3 sodium kicked out, 2 potassium kicked in. We will talk about that particular pump and how it's important in restoring a resting membrane potential for cells. All right, so I'm not going to show it here, but if you want to see more detailed YouTube video, when you're looking at the PDF version of this PowerPoint, you can click here and pull up a YouTube video on that. Okay, so we've got some more active transport to talk about. Let's go to the next slide. Secondary active transport. Okay, secondary, I think, 2 things. This is different from the sodium potassium pump. I know it might be confusing, but in this case, either a cell is moving 2 substances at the same time across a membrane, and that's called symport or co-transport because they're going together like they're holding hands, okay? Then there is antiport, where substances are going in the opposite direction to each other, okay? And that's counter-transport. Okay, so the first type of coupled transport is co-transport or symport, which means they're going in the same direction. Now here's the distinguishing thing about this. In this co-transport, energy gained from the passive transport of one ion that's moving with its concentration gradient fuels the active transport of another ion in the same direction. So the best example, and there's many, but this is the best one, is the passive transport of sodium with its concentration gradient, because sodium is always higher outside of a cell than inside, and that helps fuel the active transport of glucose against its concentration gradient. And we're going to talk about how that's important in human health shortly. Okay, so 2 things going in the same direction. The passive transport of one fuels the active transport of the other going in the same direction. The next one is counter transport. Now this is where the energy gained from the passive transport of one ion fuels the active transport of something else, like another ion, in the opposite direction. All right? Okay, let's talk about the importance of co-transport. So we talked about in the first part of this lecture, why do you give a dehydrated person who's not conscious isotonic saline? And the answer was osmosis and tonicity of solution in order to hydrate someone who can't drink for themselves to rehydrate themselves. What happens if your patient is dehydrated and they're completely conscious and completely capable of drinking their own fluids to hydrate themselves? Why would you put an IV into a patient who can hydrate themselves by drinking? Do the least invasive treatment first, and it's also the cheapest treatment versus getting an IV. Okay. Oral rehydration therapy. Okay, now I've got a picture of oral rehydration salts here. All right, it's a packet. You basically put 1 packet in 1 liter of drinking water, and it's going to hydrate someone better than just a glass of pure water. This is the essence of why Gatorade or these other, I don't want to say energy drinks, but these hydrating drinks hydrate someone who's dehydrated much better than plain water. Because if you drink something that's got a little bit of salt and a little bit of glucose in it, it's going to help hydrate them better than if it was pure water, okay? And it has to do with co-transport, which is a form of secondary active transport. Okay, so let's look at this. Chronic diarrhea, move that away. Chronic diarrhea from gastroenteritis, maybe someone's got C. diff, cholera, who knows what, inflames the intestines. It limits the ability of the intestines to reabsorb salt and water, and that can lead to dehydration, especially in children who are so small to begin with, they don't have a lot of fluids to retain, okay? However, diarrhea, chronic diarrhea, doesn't interfere with co-transport of sodium and glucose in your intestines, and this is why co-transport is important. So what happens is when salt crosses the intestinal wall into your bloodstream, okay, that passive transport of sodium is going to cross over into your bloodstream, crossing the intestinal membrane. Water follows salt wherever it goes, like a little puppy, okay? And the passive transport of the sodium fuels the active transport of glucose across the intestinal wall into the intestine. And both sodium and glucose going into your bloodstream make your bloodstream more concentrated with solutes at that intestinal wall, and water's going to follow those things into your intestines fast, okay? And that means the patient is going to get hydrated. So what we've got here is two things going on. We've got oral rehydration salts that contain salt, sodium chloride, a little bit of potassium chloride, which is also another salt, and anhydrous glucose. Okay, so diffusion of sodium across the intestinal wall into the bloodstream from high to low, passive transport. The energy saved by that is going to fuel the active transport of glucose across your intestinal wall by active transport. It's going to get into your bloodstream, salt and glucose. That salt and glucose, because it's more concentrated right now, is going to pull in the water by osmosis into your intestines, and that's how you're going to get hydrated. Diffusion, which is passive transport, active transport of glucose. This is going to be a form of co-transport, and osmosis. All right. So this is what can hydrate someone who's dehydrated. This is why we have things like Gatorade, okay? And the GIF I have over here to the left shows the transport of both glucose and sodium. Glucose is the yellow thing, sodium is the green thing, and that's pulled into your bloodstream, all right? See all the red blood cells? So this is bloodstream. Over here is intestinal membrane. When sodium and glucose are pulled in, the water molecules are pulled in. So let's look at that again, okay? This is hydration. Pulling in salt and glucose across your intestinal lumen into the bloodstream causes a hypertonic solution in your bloodstream, which pulls in water across your intestinal lumen into the bloodstream, and you are hydrated. Okay, so is this whole cell transport thing important in human health? Sure is. All right, okay. So bulk transport. This is the movement of large substances across the membrane. It's going to require active transport, and I'm going to give you several types. One is endocytosis. Endo means within, and cytosis means cell, so inside the cell. Phagocytosis is the pulling in of large particles into a cell. Pinocytosis is the pulling in of fluid particles into a cell. So here's how I remember phagocytosis. Your white blood cells phagocytize bacteria. In fact, that's why I have the GIF here. What we've got is a little bacterial cell that is being sucked into a white blood cell where it's going to be destroyed. That is phagocytosis. Now, I remember pinocytosis by thinking of, what's Pinot Grigio? If you knew that was a type of wine, then I know you drink wine, okay? But I think pinocytosis, I think Pinot Grigio, that's a liquid. That's pulling in liquid particles. Okay, now there is also receptor-mediated endocytosis where something outside a cell has to bind to a receptor, and then the cell membrane wraps around that particle. So here's the large particle. Here's receptors on the cell membrane. They wrap around the particle, pinch off, and now we've got a vesicle containing that, whatever it is. So that's receptor-mediated endocytosis. Now the reverse of this can happen with exocytosis. Here we have a large or a bunch of particles that are inside a cell. They merge with the cell membrane, and then that big, whatever it is, proteins, amino acids, I don't even know, get spit out of the cell by exocytosis. All right, so those are all forms of bulk transport. All right, we're almost to the end of this chapter. I know the suspense is killing you. Membrane potential. I gotta kind of talk about this a little bit because we're going to talk about the importance of sodium and potassium in the next chapter. And this is review of your intro biology. Okay, so the inside of a cell is, it's got, let's see, very little sodium. That's why they make the sodium symbol small. Outside of a cell, you have lots of sodium just waiting. Outside of a cell, you usually don't have a lot of potassium, but inside of a cell, you have potassium. And then a cell's got a certain number of negatively charged particles called anions. And so what happens is a cell membrane, when it's at rest, is a very negative number of minus 70 millivolts. Okay? It's resting. So the number of potassium ions that are entering or leaving the cell changes the negativity of that cell. The more potassium that gets kicked out of the cell, the more negative the inside of the cell becomes. The more potassium that enters a cell, bringing its positive charge inside, which can offset the anion concentration, the less negative the inside of the cell becomes. And a cell maintains its resting membrane potential by that sodium-potassium pump. Do you remember how many sodium leave per how many sodium enter a cell? 3 sodium leave a cell for every 2 potassium that enter a cell. That's the sodium-potassium pump. Okay, so let's look at this. Is this important for human health? It sure is, okay? And if you're looking at the PDF, this PowerPoint, you can look at the, well, let's pull it up, okay? Hyperkalemia is a high blood concentration of potassium. So hyper means excess, and the K is for potassium, and emia means in the blood. So it literally means high potassium in the blood. Who cares, right? Well, if you want your heart to stop, hyperkalemia is okay. So you really don't want that. So hyperkalemia is used to kill in the lethal injection of death row inmates. Okay, first they sedate them. Okay, they give something to stop breathing, and then they give potassium chloride to stop the heart. And they basically cause hyperkalemia that stops the heart. So if we look at the PDF, we'll give it a second to come up here. And the reason I bring this up is you're not likely to encounter death row inmates getting the lethal injection, but sometimes people who are taking diuretics, it can affect taking a diuretic to make you pee out body water to affect your blood pressure, can affect your blood levels of potassium and cause you to retain potassium, and that can cause hyperkalemia. So it's kind of important to know what that is. So resting membrane potential is sensitive to changes in plasma potassium, okay? Okay, an abnormal increase in the blood concentration of potassium is called hyperkalemia. When hyperkalemia occurs, more potassium can enter the cell. Don't worry about the Nernst equation, that's a physics thing. But what happens is that can cause your heart muscle cells to remain resting instead of contracting, and can actually stop your heart, okay? So, and notice here, for these reasons, blood electrolyte concentrations are monitored very carefully in patients with heart disease or kidney disease, okay? All right, well, let's go back to the PowerPoint. All right, so let's talk about formation of an action potential in a cell. First step, a stimulus. Something happens to stimulate a cell, and usually what that is, and think about this when we get to the beginning of the next chapter, chapter 4. A cell is typically stimulated by a neurotransmitter that's released by a neuron, and that neurotransmitter will bind to a receptor on a cell, and that will be the stimulus. All right, what that neurotransmitter will do when it binds to a receptor is it'll open an ion channel, and the ions will flood into a cell. And a stimulatory ion, one that's always stimulatory, is sodium. All right, so I want you to think about this. Here we have the skier that is waiting. Think of the skier as waiting outside the cell. And here's the membrane, and then here's inside the cell. Sodium is normally very high outside the cell and very low inside the cell. That's because sodium, if it is allowed entry into a cell, will stimulate that cell. So sodium is this skier, and it's basically at the top of a big slope. So if I move this out of the way, okay? So here's this long jump, all right? And our skier is up here at the top of the concentration gradient. And what happens is, if that little gate, that little entry, that little channel to the entry of the cell is opened, that sodium will flood into the cell like a little sodium tsunami, and it will stimulate the cell. So the stimulus is a neurotransmitter binding to a cell. It opens an ion channel, and the next step is sodium channels are opened. Sodium floods into the cell, so the skier goes into the cell, and that's going to cause the start of an action potential. Now the cell has become stimulated, it's become depolarized, it's become excited, and what happens is the membrane potential goes from a resting -70 all the way up to a positive 30. Okay, next step. Potassium channels now open. Potassium leaves the cell, taking with it its positive charge, and what happens is it's going to drive the membrane to be very negative because it's losing a positive charge. So you get this downward part of this slope of the action potential. And what happens is the cell is heading to become resting or repolarized. But the problem is, so much potassium leaves the cell that it actually overshoots the -70 and goes to around -80. That's not the right membrane potential. So the last step is going to be the sodium potassium pump is going to take in a little more potassium, kick out a little sodium to allow that membrane potential to go a little bit back up from -80 to -70. And so you get this last part of the curve right here where the sodium potassium pump kicks in. Potassium's pulled back in, sodium's kicked out, and eventually we get up to the -70 resting potential. All right, so here's what the active potential looks like. We'll go over it. What we have here, there's a stimulus, sodium channels open up here, get an action potential. At the downward part of this curve, potassium channels are open, potassium leaves the cell, but it overshoots the resting potential so that the sodium potassium pump kicks in to get it right back up to the resting potential it should be. All right, so I know this seems complicated, but I want you to remember a stimulus for a cell is a neurotransmitter binding to a receptor on the cell. When that happens, an ion channel opens up and something like sodium can come flooding into a cell and cause cell stimulation. Okay, remember that when we get to chapter 4. So what we've gone through is, well, on Monday's lecture, we talked about all of these forms of passive transport. What we talked about today was active transport and different types of bulk transport, which comes under active transport. And then we finished by talking about cell membrane potential. Okay, so this ended Wednesday's lecture. This coming Friday, I will start chapter 4, part 1. Okay, all right, so I'm going to stop the share, and here we go. We are done for today. I'll see you on Friday.